Right, let's jump straight into this explainer. Getting a handle on protein structure, from primary all the way up to quaternary, actually makes up a massive 15% of your total marks in Biology 9700, Paper 2. So yeah, you really can't afford to just skim this part. But here is the brutal truth. Year after year, we see brilliant students walk into that exam hall knowing their biology inside out, and yet they just hemorrhage marks right out of the gate.
So, why did those marks just vanish? Well, I'm going to show you exactly what the examiners are actually hunting for. We'll start with this one tiny, almost invisible mistake that costs students marks before they've even finished reading the prompt. You see, it isn't always a lack of biological knowledge that tanks your grade. A lot of times, it's just a lack of exam strategy. People just rush into explaining these super complex biological functions without laying down the basic groundwork that the Cambridge mark scheme practically demands.
Take a look at this quote right from the principal examiner report. This is exactly why your standard revision tactics sometimes fall short. Candidates consistently confuse key terminology. The fix? Always define your terms first. If you don't define your structural terms right at the start, you literally lose the foundational marks. Honestly, it doesn't matter how beautifully you describe an enzyme's active site on page 2, if you haven't explicitly defined the terminology in your opening, the examiner literally cannot give you those easy initial points.
Okay, let's build this up step by step, starting with the primary structure. Think of this like the exact sequence of letters forming a word. It's the highly specific sequence of amino acids in a polypeptide chain. And this right here is your crucial blueprint for literally everything that happens next. When you're writing this down in an exam, you absolutely have to emphasize two words: specific and sequence. Remember, it's not just some random chain, it's a highly ordered genetic translation.
Right, so once that linear blueprint is locked in, it starts to fold locally into these predictable shapes. It's kind of like biological origami. This is your secondary structure, the folding of the polypeptide chain into alpha helices or beta-pleated sheets. Now, what the examiner really wants to see here is you mentioning that these structures are held together by hydrogen bonds. That localized folding is the very first step in giving the protein its actual 3D physical form.
Then we step up to the tertiary structure. In your answers, you must emphasize that this highly specific 3D shape is what completely dictates the protein's biological function. This is a massive leap from just a folded sheet, right? We're talking about the intricate, three-dimensional folding of the entire polypeptide chain, locked in by disulfide bonds, ionic bonds, and hydrophobic interactions. Structure dictates function. So if a question asks why a denatured enzyme stops working, why a fried egg can't be unfried, it's specifically because this precise tertiary 3D structure has been wrecked.
And finally, the whole shebang, quaternary structure. This is where multiple polypeptide subunits actually interact. It's a total biological marvel, honestly. Multiple folded subunits working together as one single machine. Just picture hemoglobin with its 4 distinct polypeptide chains complexed together. Now, not all proteins reach this stage, but for the ones that do, you need to clearly state that it involves more than one folded chain functioning as a single unit.
Now, in just a second, I'm going to reveal an examiner clue that changes the game completely. Frankly, it's the difference between scraping a mid-tier grade and hitting that absolute top band. But before we get to that, you need the methodological framework that actually makes this clue obvious. Because let's face it, just memorizing definitions is only step one. Applying them to weird, unfamiliar contests, that's where the real exam actually begins. To lock down full points on those tricky problem-solving questions, you have to deploy this deconstruct, solve, evaluate sequence. It proves clear, evidence-based reasoning. First, extract the info from the prompt. Second, chop the problem into logical steps. Third, solve it systematically and, please, show all your work. Fourth, do a reality check. Is your answer actually biologically reasonable? And fifth, make justified recommendations based on the data. Cambridge awards marks for the journey, not just the final destination.
Okay, let's take a look at the top 3 examiner warnings straight from past papers. These are the classic traps that just steal easy marks from really smart students. Since these come straight from Cambridge data, you need to treat them as absolute gospel. Catch the pattern here? It is all about precision. Writing some generic waffle about protein folding when the question specifically asked about collagen in a particular context, that will strictly cap your marks. But the most painful mistake is that first bullet point. 0. That is the exact number of marks you're going to get if you forget to include units in your calculations. And yes, that applies even if your math is completely flawless. 0.
Just imagine doing all the heavy lifting, calculating the rate of an enzyme-controlled reaction perfectly, and then getting a big fat zero just because you forgot to write down milligrams per decimeter cubed. It hurts just thinking about it, but it is 100% avoidable. Always check those units.
And hey, speaking of math, when an exam throws statistical analysis at your protein data, consider this table your ultimate cheat sheet. You've got to instantly map the correct statistical test to the type of data they give you. If they hand you data comparing the mean rate of reaction of two different protein structures, boom, use a t-test. Looking at categorical frequency data, that's chi-squared. Analyzing how one variable changes in relation to another, you need a correlation test. Memorize this mapping, because picking the wrong test instantly tanks your methodology marks.
Right, let's dial up the stakes a bit and test your evaluative thinking with a real comparative analysis challenge from a past paper. I want you to play examiner for a second here. 2 answers were submitted for the exact same question, asking to compare secondary and tertiary structures. Let's see if you can spot exactly why one gets a low grade and the other bags maximum points.
So here's the flawed answer. They both fold, but tertiary is 3D. I mean, it's technically true, but it is incredibly vague. Now look at the top band answer. Both exhibit folding, whereas tertiary forms a 3D shape via disulfide bonds, unlike secondary's local hydrogen bonds. See the difference? The crucial takeaway here is that the top band student uses comparative connectives, like whereas and unlike, and backs up every single comparison with hard molecular evidence, actually naming the specific bonds. That is exactly what Cambridge means by evidence-based reasoning.
If you want to consistently score in that top band, your critical analysis has to go way beyond simple description. You need to actively demonstrate these 3 higher-order evaluative skills. Top grades require aggressive analysis, not just passive reading. You've got to question assumptions in the provided data, look at different biological perspectives, and above all, justify your decisions with hard evidence from the text or the graph. If you're just parroting the data back without evaluating it, you are leaving so many marks on the table.
And you know, this evaluative mindset completely applies to the lab as well. If you get hit with a practical experimental question about protein structures, applying this strict systematic sequence ensures you don't drop a single methodological mark. Always start by selecting the right apparatus first. Then, follow the instructions sequentially. Make really precise observations, record your data in clearly labeled tables with units, and, critically, pinpoint any anomalies in your results and suggest why they might have popped up.
Okay, we've covered a ton of ground today, so let's do a quick recap of the absolute vital takeaways. Number 1, always define your terminology, primary, secondary, tertiary, quaternary, before you even start writing an extended response. Number 2, rely on that method mark strategy to deconstruct the complex problems. And number 3, obsessively check your units before you even think about moving to the next question. Just building these 3 habits will dramatically secure your grade boundary.
Make sure to subscribe to Ace the Exams at the handle on screen so you can absolutely crush your Cambridge examinations. Because look, while you now have the structural theory and the methodology locked down, the specific command words that actually dictate your mark band, yeah, those remain a hidden trap. And we're going to decode all of that in our next explainer. Do you know how one simple verb can completely alter your entire answer? Well, I'll see you there to find out.